Hello, welcome everybody to the AOCS Student ePoster pitch competition for the Biotechnology division. We look forward to hearing the student presentations, hearing feedback and questions from the judges and the audience, and seeing participation from our live audience. I want to introduce our judges for today. They are Long Zhao, Sarah Willett, and Tom McKean. Okay, so I'll just give you a brief overview of what's going to happen today. Each of the 4 competition finalists will present for 5 minutes. After each student presentation, we'll have a 7-minute question and answer and feedback from the judges, and if there's time, audience questions as well. You can post your questions or any congratulatory notes in the chat area. After all of the presentations have ended, we'll pause the stream for a few minutes to allow for final judge deliberations. And during that time, the viewing audience will also have an opportunity to vote for their favorite poster pitch. Lastly, we'll announce the first and second place winners of the AOCS Student ePoster pitch competition for the Biotechnology division. I want to wish all the students well and thank all of our volunteer judges and the audience for attending, listening, and supporting this competition. We'll start with the first presenter in just a minute. I will go ahead and introduce the first presenter, and then there'll be a little delay while he gets his slides ready. So our first presenter is Mohammad Raihan from South Dakota State University. And the title of his presentation is Evaluating the Differences in Cellulase Activities and Proximate Composition of Different Economically Important Substrates under Submerged Fungal Fermentation. Good luck, Mohammad.
Good afternoon, everyone. Thank you for giving me this opportunity to present my study. Today's my presentation will be on fungal fermentation on different substrate, and we'll be looking on the enzyme activities. My background, in this slide, I will be talking about the background of agro-industrial residue that I used in my study and their scope and challenges. I use different kind of agro-industrial residues, and they are high in nutrient such as protein, carbohydrate, dietary fiber, minerals, and vitamins. They are readily available, cheap, and sustainable. And they have challenges. They are full of anti-nutritional factors, and they are responsible for the low nutrient availability and low digestibility. So we need bioprocessing with the goal of increasing the desirable compound and reducing anti-nutritional factor, and also look for the nutritional new value products. There are different processes have been applied to reach these goals. The microbial fermentation is one of the best potential way of getting these goals. So fermentation reduces the majority of anti-nutritional factors in most substrate, but high fiber reduction is low. In this project, they determine the proximate composition and cellulose activities using different grass microbes like Arabicidium pullulans, Neurospora crassa, and Trichoderma ricei, and cultivated on 3 different substrates under submerged fermentation. Our process was very simple: substrate, fungi with water, and then get the improved fermentation product with low anti-nutritional factor. The hypothesis of my study would be as low cellulose activities are responsible for low fiber reduction. And the scope of my study will be understanding all the cellulose activities and designing the optimal fermentation process for maximal process efficiency. In this slide, I'll be talking about the materials and methodology. It was very simple, and we used 3 different substrates: the dye-processed pea protein and the ULP, and ethanol industry byproduct DDGS. And in this fermentation, we used 10% solid loading rate and inoculated them with all 3 microbes, Apollons, Encapsulation, and Tiericea, and then incubated for 120 hours. We separated after 120 hours. We separated them in a liquid and solid fraction. Then they are liquid fraction was analyzed for cellulose, beta glucosidase, and endoglucanase activities. And then they were oven dried and analyzed for crude protein, mass balance, and also in solid fraction, saponin, total phenolics, crude fiber, and ash. In this slide, result slide, I will be presenting my data. I'm not presenting all of my data. Here I have my substrate, DPP, DHP, and DDGS, and in the process I have raw, control, and all 3 microbes, and then total solid recovery and crude protein in solid fraction and crude fiber, and then my enzyme activities. As you can see here, total solid recovery was low with Encapsulation in all 3 substrate in solid fraction. That might be the reason that they solubilized or like microbial conversion was higher, and they ended up in the liquid fraction. The crude protein was higher with the Tiericea and in all 3 substrate, and they followed by Encapsulation and Apollons. The crude fiber was not degraded in with Apollons and Tiericea, and there was a very minimal degradation with Encapsulation. In the cellulose activities, we saw that there was very low cellulose activities in all 3 substrates with all 3 microbes compared to the literature, which could be like 3 to 50 unit per gram. And beta glucosidase activities was high with Encapsulation in both the DPP and DHP. The endoglucanase activity was similar, but it is also low compared to the literature. In the summary and conclusion, all my substrates that were inoculated with the inoculated with the microbes, they increase the protein content, and Encapsulation in terms of high solubility and enzyme activity was most effective, and low fiber degradation was caused of the low cellulose activity. That was my hypothesis, and in future we will be working with like more substrate for better understanding, and we'll also look for a different time point and introduce co-culture and solid state fermentation and compare my results with this result, and then this will give us the idea of making a better fermentation process with better efficiency. Thank you. I'll have question now.
Thank you so much, Jahir. And now we have the opportunity for the judges and the audience to present their questions. To the audience, please put your questions in the chat box. And if there's time after the judges have their opportunity, then I'll read them out loud. So go ahead.
So Mohammed, this is Joe, great presentation. So do you mind help me understand better in terms of your background and the rationale, why you want to remove those attention factors? So what's your target application? It's because that is important for your job here. And also the second one, you mentioned about some of the phenolics as well as saponin as your attention factor. Do you mind to elaborate a little bit more why those two will consider as the attention factors?
Okay.
And the third one, I'll be quickly here. Do you mind to tell a bit more about your conditions for your current approach, submerged fermentation? For example, I didn't read clearly about your time, temperature, how long you run it, and the pH, etc. For those things. Would you mind to give me more detail for the audience?
Okay. So thank you for your questions. And if I hear you correctly, that your first question was that why I used like this fermentation process and why I look for the anti-nutritional factors. So anti-nutritional factors like different anti-nutritional factors that need to be removed for like better use and use. Like if we talk about the saponin, saponin in my substrate, I use two substrate, DPP and DHP. They have saponin in them, and they have a very big market here in USA. But their limiting factor is their smell and off flavor, and that is saponin is attributed to that, contribute to that off flavor and smell. So, and also we use the submerged state fermentation, and the pH was set for the microbes, and it was like 5, and we continued it for like 5 days. The fermentation was 5 days, and it was 10% solid loading rate, and yeah, we use 10% solid loading rate then, and we autoclaved it, and then we inoculated with microbes, and then we put them in a shaker for like RPM was 150, and then carried out the fermentation for 5 days before processing.
Do you need the air? Or you don't need the air?
No, we don't need air.
Since you mentioned about saponin, just as an example here, have you measured that after your fermentation process, or you have not measured them?
No, we did measure them. Like we did measure the saponin, but in DPP and DHP, the saponin was concentrated in solid fraction. Especially with the DPP, with Encapsulation, it was increased a lot.
Okay, I turn over to other judges for your questions or comment.
Very nice job to hear. I really liked the use of your method slide. I thought it was a very good visual. My question for you is more so on your substrates. How did you go about selecting the substrates? And did you look at different substrates at all? For example, different particle size, or why are you looking to decrease the fiber content?
So yeah, like with all, we selected this substrate because of their economic value, and we are looking for other substrate too for my future studies, and like we didn't look for the particle size, but their composition. Like one of my substrate DPP was high in protein, and DDGs was lower in protein, like it's 32%, and DHP is even lower. So we took these 3 substrate, and then we go for the cellulose activities. With all the other studies, we also saw that like the other anti-nutritional factor can be decreased easily, but the fiber is always remain high. So as these substrate are already in high demand, but they're not being used, they're underutilized, and we wanted to see like whether the fiber degradation is done by the enzyme estimation or enzyme activities.
Yeah. And then my second question is, what other methods are you going to look at to increase the cellulase activity? Like, is there a pre-treatment or other things?
So that's a very good question. So my future study will, I will be looking for the solid state fermentation. We know that in the solid state fermentation, the enzyme activities are usually higher. And then I will also look for like adding the cellulase, like after the autoclaving, and the supplementing the cellulose with might help the higher fiber degradation.
Hi, Jahir. This is Tom.
Hi, Tom.
Very good talk. How do you think this fermentation method compares to, say, physical fractionation of these different substrates?
Okay, so during the physical fractionation, usually the dry fractionation or air classification, when we are concentrating the protein, sometimes we are seeing the contamination is high. But when microbes are converting them, then it is possible to get the better end product.
Okay, thank you.
Thank
So
you.
I just add on more. I don't know, Jill, we have more audience questions from the public.
No, I don't. Go ahead. I was just going to say I don't see any audience questions, so go ahead if you have another one.
Yeah, I just come back to the original question. So just want to clarify, what's your final target application? Are you going to target on the feed side, like a poultry swine, or you target on the food side for applications?
Okay, so these are like, we will go for the feed side first, and then we will look for the food side then, if everything met, like all the criteria are met, and then we will go for the food side. And for DPP and DHP, definitely we want to go for the food side.
Okay, so in the future, I will looking forward to more on your final validation from your application side. So I understand better, no matter from the protein functionality, as well from feed side, how the fermentation does to the animal behavior and the performance by feeding your ingredient, because that's important on that side. And additionally, I would like to know a little bit in the future work. My comment is you are still working on a little bit high premium ingredient compared to we call the lots of the residue. No matter US, you know, we have lots of corn and soybean, we have lots of sorghum. I don't know for that side, if you think about anything we can utilize more, add more value on that thing, will help industry better. So
yeah, we will look for that the corn, but like in the staying in the middle Midwest, and we can you know, like we have a lot of DDGs is being produced for the ethanol factory, so we can utilize that too. So we will look for the other substrate, but like this is my primary study, and then we will extend further for the other substrate.
Okay, great. Well, we're all out of time. Thank you so much, Jahir.
Thank you everyone.
Yeah, thank you. We'll move on to our next speaker in just a moment. Okay, our second finalist is Madison Oehler. Madison is a master's student in biosystems and engineering at the University of Tennessee in Knoxville. She's finishing her thesis on the encapsulation of antimicrobial peptides in bicontinuous microemulsions for topical delivery. The title of Madison's presentation is Encapsulation of Melittin in Bicontinuous Microemulsions for Topical Delivery. Good luck, Madison.
Thank you for that introduction. Surgical site infections, chronic wounds, and antibiotic-resistant microorganisms have led to an increase in medical bills and patient discomfort each year. Antimicrobial peptides, or AMPs, are a possible solution for a novel treatment. AMPs are naturally occurring oligopeptides that target and disrupt cell membranes, seen here in this diagram, of unwanted hosts making them difficult for microorganisms to build a resistance to them. Although AMPs sound like a simple solution, few treatments have been able to make it into the pharmaceutical industry due to the difficulty in encapsulating and effectively delivering them. This project proposed the use of bicontinuous microemulsions, or BMEs, to effectively encapsulate and potentially deliver AMPs. Microemulsions are thermodynamically stable mixtures created by combining oil, aqueous solution, surfactants, and sometimes a co-surfactant. They are optically transparent, dynamic in their nature, and surfactant structures that resemble elongated nanodomains with low interfacial tensions. So AMPs, which are typically cationic and amphiphilic in nature, are attracted to anionic cell membranes. We hypothesized that BMEs created with anionic surfactants would successfully encapsulate AMPs, transforming them into their most active state because of electrostatic forces, resulting in an effective treatment. We successfully identified 3-phase and 1-phase BME systems created with biocompatible components, 2 of which are pictured on the right of this slide. For AMP addition, we chose melittin, which is an alpha helical AMP, and that originates from bee venom. It shows functions against bacteria, fungi, and viruses, and we were able to successfully encapsulate melittin into these systems at concentrations of 0 to 2 grams per liter. And for our 3-phase system specifically, we achieved greater than 98% extraction from an aqueous environment to the middle bicontinuous phase. Once these systems were able to successfully encapsulate melittin, we then decided to characterize the BMEs and the AMPs within those BMEs. So starting with small angle X-ray scattering, which was used to determine the effects of melittin on the BME structures, the data we gathered was fit with the tubular strain model, which is shown on these 2 graphs on the right. And the bell-shaped curves that you see on these graphs are typical for BMEs, which we can confirm that on a molecular level, our systems are bicontinuous in their structures. From that fitting, we were then able to determine the quasi-periodic repeat distance, which is the distance across the oil and water nanodomains within the BMEs. And if we follow down to this table, as the melittin increases, the repeat distance decreases, which tells us that the melittin is reducing the surfactant's efficiency at solubilizing oil and water. Then to characterize the melittin, we use circular dichroism to determine the structure within the BMEs, which measures ellipticity. A typical spectra for an alpha helical peptide has 2 minimums at 208 and 225 nanometers. So on this graph on the left, highlighted at 208 and 225 for the blue and red lines, which represent melittin in our BME systems, they mimic the alpha helical spectra. When we compare that to the black line or melittin in an aqueous solution, there's only 1 minimum. We can conclude that melittin encapsulated within our BMEs, as a higher percentage of it is more active than when it's in an aqueous solution. If we follow to the right graph, the fluorescence, we are looking at, again, the black and the blue and red lines at the maximums, and that shift to the left, or a 20 nanometer blue shift. This tells us that the melittin is residing in a more apolar environment within those surfactant monolayers. And then finally, disk diffusion and well diffusion are currently being tested for the effectiveness of the AMP loaded BMEs. And this is done by creating lawns of bacteria on an agar and then adding a treatment either to a well or to a disc and allowing that to incubate for 24 hours and checking to see if there is a zone of inhibition, which means an absence of bacterial growth due to the treatment that was applied. So in our initial testing shown in this left picture, we were struggling with mass transfer issues due to the BMEs and the agar solutions. But in the right 2 pictures, we have been seeing some success in using this newer media, showing some inhibitory effects of our AOT tween 85 systems with the incorporation of melittin. And I'm excited to continue with that research to make those final conclusions. But with that, I would like to thank you for your time and would be happy to answer any questions that you have.
Great presentation. It's now time for the judges to provide feedback and ask any questions. And I'll remind the audience, if you have any questions, please put them in the chat box.
Very good talk, Madison.
Yeah.
So melittin is a bee venom, and I mean, is that a major allergen in bees or minor?
Allergen, you mean to humans or
To humans, yeah.
Yeah, so I've got a question before.
Sorry, I'm still not.
Oh, sorry, I'm here. Yeah, if we can use animal components. So they have incorporated other types of AMPs that were from animals in their treatments, and they haven't shown too many, I guess, allergic effects. So melittin's one of the most studied AMPs known to the state, and so that's why we chose that as our first AMP first. And so I haven't seen too much on that, I guess, any on allergens specifically.
Okay,
Thank you.
thanks.
Hi, Madeline. Madison. Great job on your presentation. I did have a question more so on the production of the BMEs. Could you explain more about the conditions? Did you have to optimize any conditions to produce these? And also, have you looked at the stability of them?
Yeah, so there are different types of microemulsions, and we chose bicontinuous specifically because they are more stable and they are quicker to form. I guess the quicker form is a better answer to that. And then they have those elongated nanodomains. So these are typically, the way I'd make them is creating my aqueous solution that will have the melittin in it. And then I create a solution that has the surfactants and the oil combined together, and then add that aqueous solution to that and gently mix them to not disturb the AMP. And then, so we did have to optimize. These surfactants can be highly affected by temperature and salinity. So when we incorporated melittin into these systems, we did have to change the salinity of the system so that they weren't shifting maybe to a, I don't know if you're familiar with a Windsor 1 system to a Windsor 3 microemulsion system. But for the systems that we did have in this project, they were able to be stable at room temperature and created at room temperature, and then up to the incubation, which is 37 degrees C or skin temperature. Thank you
so much.
So Madison, I have two little questions for you. So the first one is regarding, because eventually you talk about this is for topical delivery. So when you select surfactants, is there any consideration which one you have to choose specifically? And the second question is regarding when you talk about is a bicontinuous, what do you mean for that? And also you talk about microencapsulation, how you evaluate you have a well encapsulate or not? How you do that, no matter physical, chemical, way you do that? Would you mind to elaborate a little bit more on these two questions? Thank you.
Can you repeat that? Or I could start with bicontinuous first. Like I forgot the first one, but okay. So bicontinuous, what that means is that the microemulsion, the surfactants in the microemulsion are solubilizing equal amounts of oil and water together, creating, instead of, if we think of other types of microemulsions, it's droplets of oil and water or water and oil. And so with bicontinuous, you have these kind of moving elongated nanodomains that kind of act as maybe a mimetic biomembrane for the AMP to reside within. Instead of, if we had droplets, there wouldn't be an area for it to reside in, I guess, if that answers your question, hopefully. Not to go too far into detail on bicontinuous. And then, sorry, what was your first one?
My first one was regarding whether, because go to topical, there's lots of regulatory regarding which ingredient can be used, which one cannot use. You any consideration on that side regarding you choose your surfactant on that side? Thank you. And as well for your oil system, which oil you choose, right? If any consideration on that side?
Yeah, so we specifically for this project, we're definitely looking into biocompatible components because there has been research with previous components that were able to successfully encapsulate proteins and AMPs, but they did not use components that could be applied to the skin. And so the ones from at least literature review and the systems that I ended up working with, those were ones that people have used. They added, it was more drugs, not peptides into those systems, but they were biocompatible. So then moving forward, if this were to work, then we could eventually apply them to the skin. Also, I do want to add too, if they would be a topical treatment, there's this talk of adding some type of either a thickener or a gelatin to make it more of a gel, consistency to deliver it more effectively as well, because it is liquid right now. Thank you.
Okay. Any question from the audience?
Not seeing any questions from the audience at this time. Do the judges have any more questions? Otherwise, we can move on to the next presenter. Okay, thank you so much, Madison, and we will move on momentarily. All right, we're back with our third finalist in the Biotechnology Division's ePoster pitch competition. Our finalist is Eugen Lee. She's from the Department of Integrated Biomedical and Life Sciences at Korea University. And the title of her presentation is Novel Strategy for Synthesis of Stearidonic Acid-Enriched Triacylglycerol from ahi flower seed oil via a two-step enzyme reaction. Good luck.
Good afternoon, everyone. This is Eugen Lee, and today I'm presenting the preparation of stearidonic acid-enriched triacylglycerol from ahi flower seed oil via Candida rugosa lipase catalyzed hydrolysis and Aversa immobilized lipase catalyzed esterification. First of all, stearidonic acid, as known as SDA, is C18-4 delta 6,9-12-15, which is one of the omega-3 fatty acids as plant origin. This is the structure of SDA. SDA has multiple benefits on human health for those who have high cholesterol, heart disease, and diabetes. And in this study, I use ahi flower seed oil as a substrate because it contains SDA a lot and is non-GMO. I chose to enrich SDA because it is better than other fatty acids in our body. SDA is biosynthesized from ALA by a limited enzyme, a delta-6 desaturase. So the conversion rate of SDA to EPA is higher than ALA. When only 2 to 5% of ALA is converted into EPA, 20 to 30% of SDA is converted. My experiment is mainly about synthesizing SDA-enriched TAG via two lipase catalyzed reactions, which are hydrolysis and esterification. For the first step, SDA is enriched into glyceride fraction from ahi flower seed oil using Candida rugosa lipase catalyzed hydrolysis. Next, SDA-enriched TAG was then synthesized from the SDA-enriched glyceride and the SDA-enriched fatty acid via esterification using a biocatalyst, the Aversa immobilized lipase, the in-house enzyme. The two main key points are that I use glyceride instead of glycerol to synthesize TAG, and I use Aversa, the in-house immobilized lipase. This scheme shows the overall procedure. The first step is the SDA enrichment in the glyceride fraction, and the SDA-enriched fatty acid was obtained from the part of the SDA-enriched glyceride by saponification. Next step is the synthesis of SDA-enriched TAG. For the method, both steps were carried out in the flat-bottom glass vessel with a water jacket. Moreover, the second step, esterification, was carried out under the vacuum condition. This is the picture of the vacuum gauge monitor, the vacuum bell, and the vacuum pump. Moreover, the Aversa was prepared from liquid Aversa transform 2.0 using Willett BPOC 1600 as a carrier. Carrier was pre-soaked with ethanol and combined with enzyme solution with buffer. After water basting for 17 hours, it was washed with the buffer solution and dried. This lipase is developed for the production of biodiesel. As a result, in the first step, the optimum conditions was 35 Celsius with 0.1% enzyme loading, and SDA was enriched into glyceride fraction from 21.6 to 40.7%, which is twice more. In the second step, the optimum condition was 50 Celsius with 10 torr. A maximum yield of about 94% was achieved after 12 hours under the optimum condition. In conclusion, SDA-enriched TEG was effectively synthesized by two lipase catalyzed reactions, hydrolysis for the enrichment of SDA in glyceride fraction, and esterification for the synthesis of TAG. This study presents a novel strategy for the synthesis of omega-3 enriched TAG using in-house immobilized enzyme. Special thanks to Professor In-Hwan Kim, and this was the end of the presentation. Thank you for the listening.
Great job, Eugen. We now have time for audience and judge questions. I'd like the audience to please put your questions in the chat box. And go ahead, judges.
Hi, Eugene. Very nice talk. Thank you.
Thank you.
So ahi flour was developed by breeding. Is that correct? Is it,
Yes. Like in our lab or?
No, I know it was developed by a company. I can't remember the name of the company. But,
Yeah, that's right.
Is it commercially available?
Yes.
Okay, and trying to think, when you've modified, made these triglycerols through this modification method, are they allowed to be consumed, or are they okay as supplements, or how can they be used?
I think it can be used in the commercial way and in the industrial field, but I haven't tried and I haven't think about using this as in the industrial field. So I think further study might be needed for that.
Okay.
Thank you.
Nice job, Eugen. I had a question regarding the scale. What was the largest quantity that you produced of these structured triglycerols?
For the large scale, you mean for the hydrolysis or esterification or the entire thing?
Yeah, what was like the final quantity that you got for your final product?
It was like 5 grams.
And are you looking to do larger scale production of these? How would that look like?
I think I can do that.
And can you explain more about why vacuum was used for the esterification?
So it is well known that water is not good friend in esterification with lipase enzyme. So the reverse reaction might happen. So if water isn't removed efficiently during the esterification, the hydrolysis reaction will occur instead of the esterification. So if so, the synthesis of TAG will not occur or it will be decreased. So that's why it was performed for the vacuum conditions.
Thank you.
Thank you.
So Eugene, I may have several questions here because this is a great talk. So The first one is regarding one of the enzymes you have used, because the first one is very well-known, Candida rugosa, but the second one is a commercial enzyme because you do the immobilization, or this is a regular 1,3 specific enzyme, or is a specific priority design you guys, your lab have developed? Could you mind give a little bit more details on that?
Yes, I can talk more about the Ebersa. So Ebersa is the immobilized lipase, and it has no difference in activities compared to Novozyme 435, which is known as the most effective and also expensive lipase for synthesizing TAG. And it is made in our laboratory with chip source enzyme, the liquid Ebersa lipase. So it is way cheaper than the other commercial available lipase. And I could not put the screening trial for commercial immobilized lipase and Ebersa in this poster due to the limited space. However, a screening trial using some other commercial immobilized lipase such as Novozyme 435, Lipozyme RMIM, and TLIM with Ebersa was conducted for the study. And as a screening trial result, Ebersa showed no difference in activity compared to Novozyme 435, as I say. So it was chosen for this study due to its effectiveness and economic feasibility.
Okay. And the second question, I'll go very faster here. What's the enzyme loading you are using for each of the reactions here? How much % of the enzyme you have used?
So you mean the optimum conditions?
Yes.
So for the hydrolysis, the optimal enzyme loading was 0.1%, and for the esterification, it was 10%.
10%. So my following up question on this one is maybe a little bit challenging for you, because as you know, ahi flour, as Tom mentioned about, is not cheap raw material here, and you are doing further concentration here, and you are using two step here. You have lots of enzyme, lots of the processing here. So I don't know whether in your idea you have anything. First question is why you want to do concentration, okay? And the second one is there any economic sense in your mind for the final product after you concentrate that one? Have you think about that in the future? Thank you.
So first,
The first one, yeah, let me translate it simply for you. The first one, why you want to do concentration? And the second one, what's the economic value for your approach?
For the economic approach, I know flower seed oil is expensive, but I chose it because that one is the non-GMO wild type seed oil, which contains the SDA most, like about 20%. And I have tried other substrate like equum oil in the other studies, but the flower seed oil has the more SDA content, so I chose it for the academic way. So I'm not sure about that for the economical way. And for the other question, I want to enrich into the TAG form because like there is the other form like ethyl ester or fatty acid form to enrich the fatty acid, but I think the TAG form has the higher oxidative stability and it is also in the natural form than other forms. So I think it has good stability. So if we enrich the fatty acid into the TAG form, I think it's good for the human health, for the nutrition supplement. Yes, and that's all. Thank you.
Great, thank you so much. We are all out of time for questions. We had one question from the audience, but it sounds like you answered it in your response to one of the other judge questions. So we'll move on. Thank you so much and great job, Eugen.
Thank you.
Okay, our fourth and final finalist in the Biotechnology Division's ePoster pitch competition is Mi-Soon Park. Mi-Soon is studying for a PhD in integrated biomedical and life science at Korea University. And the title of the presentation is Synthesis of Phenolenic Acid Enriched Triacylglycerol from Pine Nut Oil using or via a two-step consecutive enzyme reaction. Good luck, Mi-Soon.
Thank you for introduction. This is Mi-Soon Park from Korea University. Today, I'm going to talk about the concentration of phenolenic acid. My research title is Synthesis of Phenolenic Acid Enriched Triacylglycerol from pine nut oil via a two-step consecutive enzyme reaction. Now, I will start my presentation. Phenolenic acid, PLA, is delta-5 unsaturated fatty acid. PLA is unique fatty acid which found at pine nut oil only. PLA has a positive health effect. In particular, it may function as an apoptite suppressant through its effect on satiety hormones. Objectives of this study is synthesis of phenolenic acid enriched triacylglycerol. Thus, PLA enriched TH was synthesized by two steps like this. In step 1, we enriched PLA into ethyl ester fraction from pine nut oil via lipozyme 435 catalyzed ethanolysis. In step 2, PLA enriched TH was successfully synthesized with PLA enrichment from step 1 and glycerol via lipozyme 435 catalyzed esterification. This slide is the overall protocol for synthesis of the PLA-enriched TAG. This protocol is composed of two steps. In step 1, we obtained PLA enrichment as ethyl ester form from pine nut oil by lipozyme 435 catalyzed ethanolysis. The PLA-enriched ethyl ester from reaction mixture was isolated using molecular distillation. In step 2, PLA-enriched TH was synthesized via lipozyme 435 catalyzed esterification under vacuum. In this step, two different types of acid donors, such as PLA-enriched ethyl ester and PLA-enriched fatty acid, were used. The PLA-enriched fatty acid was obtained from ethyl ester by saponification. Now I'll explain the result. The first thing is step 1, lipozyme 435 catalyzed ethanolysis. The initial PLA in pine nut oil was 14%, and it's concentrated to about 41% in ethyl ester fractions after lipozyme 435 catalyzed ethanolysis. So we concentrated PLA about 3 times. This slide shows reaction scheme of lipozyme 435 catalyzed esterification for synthesis of PLA-enriched TH. This slide is the result of step 2. First is effect of temperature and acid donor. Two different acid donors such as ethyl ester and fatty acid were used. As you can see, higher TAG conversions with fatty acid as acid donor were achieved compared to ethyl ester at all temperatures except 40 degrees Celsius. 60 degrees Celsius was selected as an optimum temperature considering equilibrium time and cost of energy. Second is effect of vacuum on the right side. No significant difference in the conversions between 1 and 50 torr was observed. So we selected 50 torr as an optimum vacuum, and above 95% conversion was obtained as maximum. This slide shows fatty acid compositions of TH synthesized at different reaction types. PLA content in TH synthesized at 2 hours was slightly higher than others overall, but the difference is small. Finally, to summarize this study, the PLA-enriched TH was synthesized by a two-step lipase catalyzed reaction from pinot oil. And I would like to thank Professor In-Hwan Kim for their sincere guidance. Thank you for listening to my presentation.
Great job on your presentation, Mi-soon. We now have 7 minutes for questions from the judges and the audience. Audience, please put your questions in the chat box.
Good job, Mi-soon. I had more questions on your first step, the ethanolysis. Can you describe more the conditions that you use, such as substrate to ethanol ratio, enzyme quantity, things like that?
Ethanolysis and esterification. First, ethanolysis is we used the pinot oil 5 grams, and we use ethyl ester as substrate, and mole ratio is 1:50 to total substrate weight, and our ethanolysis experiment was 55 degrees Celsius, and our maximum time is 6 hours.
Thank you.
Thank you.
So, Min-Shu, I have 2 little questions for you. The first one, maybe I missed that. Would you mind retell again, what's the panolenic acid level in the final TAG form? Although you mentioned about yield, 95 the yield, but what's the concentration in that tag molecule?
We concentrated PLA 41%.
You said 41% in the 41%? Okay. My second question is because I'm aware your lab, Dr. Kim, have run lots of the trials and work on the same strain. So question for you: what's the unique? What's the new for your study here?
My first, PLA is functional fatty acid that helps our health, and all the other PLA studies so far has ended at the concentration stage. However, unlike our other studies, in our study, the concentration stage and the synthesis of TH were carried out at the same time. I think this is the advantage of this study. And it is one step further than others. In addition, the difference between acid donors was compared during the study, and the TG form, which is a natural form with high oxidation stability was synthesized as a final product. I think this is also an advantage of this study. Thank you.
Thank you.
Hi, Misun. Very good talk. Is there another way to separate the ethyl ester from the glycerides? So instead of molecular distillation, is there another way that it could be done?
We actually, I think we need more studies to answer this because, but I think molecular distillation is very best method. But we need to further study next time.
Okay, we did have one question from the audience. I'm not sure if you can answer it quickly or not, but the question is how to concentrate PLA from pine nut oil by ethanolysis.
According to previous study, most of the PLA in pine nut oil is located in SN3. Typically, like lipozyme 435 does not have radiospecificity for TAGs in hydrophobic circumstances, such as oil or hydrophobic solvent. However, in environment circumstances with an excess amount of ethanol, lipozyme 435 exhibits strong radiospecificity toward SN3 or SN13. So, using this characteristic of lipozyme 435, we concentrated PLA from pine nut oil by lipozyme 435 catalyzed ethanolysis.
Okay. I don't see any more questions from the audience, so we'll take a brief break but stay on because after this we'll launch the audience poll and the judges' deliberations. Thank you very much, Misun. Great job. All right, we have all of the judges and audience scores tallied, and it's time to announce the winners. The first place winner will receive a $200 cash prize, a recognition certificate, and a complimentary 2023 AOCS student membership. Our second place winner will receive a $100 cash prize and a recognition certificate. So with that being said, I first want to congratulate all of the finalists today, because you all did such a great job, and I'm sure it was really hard for the judges to decide. Our runner-up is Eugen Lee for her talk entitled Novel Strategy for Synthesis of Stearidonic Acid-Enriched Triacylglycerol from ahi flower seed oil via a two-step enzyme reaction. So congratulations to Eugen.
Congrats.
Very good.
Congratulations.
Thank you so much.
All right. Our first place winner is Madison Oehler from the University of Tennessee for the presentation Encapsulation of melittin in bicontinuous microemulsions for topical delivery. Congratulations, Madison.
Thank you.
Excellent job.
Congrats.
So I want to thank all of the judges and our audience for participating. I want to thank all of the finalists again for your great presentations. We look forward to seeing them again at the AOCS annual meeting. And thank you all for voting for your favorite poster pitch.
Thank you, Jill.
Thanks, Jill.
Yep.
Jill.
Thank you, everyone.